So we're going to move on to our next talk from Professor Jason Ritt from Boston University. We'll continue on the theme of controlled neural circuits. Jason?
I think I have a way to get the wires out of the way. So this is a brain-machine interface. All right. Well, thank you to the organizers. Thanks for all of you for being here. And this is a neurocontrol talk in a neurocontrol session. But that really means a lot of different things, that term. And one of the things I want to try to do here is be clear about similarities and differences. But I guess a punchline right from the beginning is even the formulation of what we mean by neurocontrol is extremely important. And I think we probably need to be more precise in what we're describing and maybe put a little bit more thought into what that model formulation ought to be. So in my case, I want to start by describing so the experimental work in my lab as a sort of context motivating the particular choices we made, some of which are going to be very similar to Garrett, some of which are going to be quite different and have different issues of tractability and all that. So I also work in the Whisker system. Garrett and I have been colleagues for a long time. This is high-speed video taken from under the animal. So you're watching them cross this track. It's slowed down by about a factor of 20. So this is going much, much faster in real time. And basically, he's searching for this water reward inside this little aperture. If you zoom in on this part here, you see he's searching, and it's under infrared illumination, so he's in the dark, but he's moving these whiskers, will eventually make contact, and then say, oh, now I know there's an object there, and I can go in and get my reward. So this is this fantastic model for active sensing. That's really the thing that we focus on a lot, which is sensations of behavior. You go out in the world and you take information from the environment. And so we think about that as the sort of closed-loop thing happening within the brain. The animal moves its whiskers, gets some information that somehow is encoded neurally, and that will drive the way it explores at the same time that it's driving whatever the animal perceives. So in particular, it can be changing its motions to get different kinds of information. And we definitely observe that in animals. There's been a lot of work on that by many labs. In our case, though, we want to understand this sort of sensory motor loop a little bit better by adding this artificial feedback channel. Can we disrupt the information as it's coming into the brain and watch the way that changes how the animal searches? So in terms of instrumentation, what that means is we measure whisker motions by measuring the whisker muscles. We do some processing on that basically allows us then to fire a laser into their brain using the same kind of optogenetic stimulation that Garrett so helpfully just described. So here's an example of that in our case where we make these, we have some papers out on these particular customized version of what's known as a hyperdrive, which is a bunch of different electrodes we can move and a fiber running down the middle. As demonstration here, this is a mouse with a genotype that's called PVCHR2, and all that means is that it's inhibitory interneurons that become active when we fire the laser. So you can see if we take over 1,000 trials here, fire the laser, here's a bunch of different spike times in this raster, and they all go away shortly after the laser. So we are lowering total activity by activating neurons that inhibit their neighbors. Here's a different genotype of mouse, the I1CHR2, where shortly after firing this laser on many, many trials, we get an increase in activity. Right? So here's the basic ideas of why one wants to use optogenetics. You have a lot of control over cell types and so forth. One point I want to make right now that's, I believe, Gary, those were head-fixed animals in your case? Yeah. So there are differences that may not seem like a big deal, but they're enormous. Everything that I just showed you, I started with the video. These animals are running around, right? So as opposed to Garrett didn't show you any examples, but his are bolted in place. That radically changes what you can use to measure the brain and also what you can use to stimulate the brain, right? That's why we have to make these custom hyperdrives. That also means that our observability is even worse than what the observability could be given current technology, because we have to fit everything inside something that's about 2 grams, fits on the animal's head, and they can run around totally unconstrained, right? If we didn't do that, we couldn't look at the active sensing. They wouldn't be able to make those behavioral choices. Right? So that'll come back a bit when we get into the control proper, but just to give you some sort of taste of why we do all this, is that now, that artificial loop that I was talking about, we can look at their whisker motions, given in red, and we can fire the laser according to the motions they're making. So not entraining them to anything. We're not picking the spike time target like in the talk you just saw. We're disrupting their natural behaviors. So here again, if you see in red, you see these laser pulses are occurring on the leading edges of these whisker motions. Here, the pulses are occurring on the trailing edges. So basically, we're stimulating either when the whiskers are moving forward or where the whiskers are moving backwards. right? And so there we can look for different sort of timing issues. And one reason why you might just imagine this is going to be important is because if I'm reaching out in the world trying to get information, I expect most of that information to come in as I'm moving forward, not as I'm moving backwards. So is there any evidence for that kind of processing where on tens of millisecond scales, the animal is responding differently to incoming sensory information based on its own motion? Those are the kinds of questions that we ask. And to give you an example of that, there certainly are differences that matter. So this is a bit of a busy slide, but I'm showing you basically in black here spike rates locked to, I don't know if you can see at the bottom, the time that we fire the laser pulse. So I've just aligned a whole bunch of laser pulses. And if we focus in on this bit right here, in the top I'm showing you what happens when we stimulate while the whiskers are moving forward, and you get this nice large increase in spike activity that actually goes off the top of this figure. But if I wait about 20 milliseconds or so, give the same laser pulse, but just wait until the whiskers are moving backwards, I get a much suppressed, disrupted response. So on tens of milliseconds, the cortex, where I'm stimulating here, the somatosensory cortex, responds entirely different to stimuli based on where the animal is in this regular sort of 50 millisecond scale motion. Right? So there's some kind of cyclic excitability that if you sort of take that to an extreme, says the animal selecting sort of the importance or meaning of a spike based on what it's doing at the moment that spike comes in. Those kinds of issues are the things we want to understand better. But so now I'll move away from the details of the sensory systems neuroscience and try to get to, okay, so how does that then motivate the kind of control problems we think about? And so let's think about what we're actually doing. Everything I just showed you was really just temporal disruption. It's not like we had a target spike rate we were aiming for. We're trying to disrupt the system as the animal's moving, and the one variable we can actually control reasonably precisely is timing. Not so much the size of the change in neural spike rate, but just when does that disruption occur. If you look in the brain, so this is a coronal slice through one of our mouse brains, at all the cells that respond to light, so this is a Thigh-1 animal that I'm showing you, all this fluorescent labeling out here in cortex, these are all neurons, neuropil, you're seeing dendrites and axons, that will all respond every time we shine the light. And in fact, the rest of the brain lights up pretty well too. So this is not a super precise stimulus where we're hitting 1 or 2 neurons. In fact, we're hitting probably hundreds to 1,000 or maybe even 2,000 neurons at a time. This is a very large stimulus. If you look around our electrodes, again, sort of classic picture, electrode goes in the brain, well, there's no empty space. It's going to be surrounded by different neurons. And that would also be true if I was trying to do electrical stimulation. I've sort of got something order of 100 or so neurons that I have to contend with. So when we think about what kind of interfaces are available, Garrett nicely set that up, talking about a lot of the interfaces are, we wish they would be better. That's a little bit true on the electrode side. So people are using arrays like this. We can get 100 or 1,000 contacts now out of these. So maybe that helps with variability, but the thing you got to keep in mind is this is a 2 millimeter scale device, meaning there's millions of neurons in the volume that this electrode goes in. So having 1,000 electrodes in a order million size network is still pretty radically underactuated and underobserved. And when you're using light, like we are, you're sort of hitting a bunch of neurons all at once. Some are light sensitive, some are not. So when we think about what kind of controllability we want, we have to think about, well, what are our objectives? If we're doing bulk problems like deep brain stimulation, something like that, where we just want a sort of overall state of the brain, well, we've done a pretty good job making a lot of progress on those. We're just asking, we want you to not be epileptic, right? But the kind of problems that we're talking about here, say something like artificial sensation, I want to somehow understand the sensory system, requires a bit more either luck or information. So one of those things that's important so far in all the examples I know of from people experimentally changing percepts relies on topographic organization. I'll give you a bit more sense of what that means. Here's a schematic where, let's suppose you can get 2 electrodes reasonably close to each other, and it just so happens that a bunch of neurons under one electrode have sort of an A tuning, whatever that means, maybe whisker deflections forward, and in some other column, a bunch of neurons happen to be similarly tuned, meaning they're responsive to whisker deflections backwards. If you're in this situation, you're probably pretty good, because if you want to bias towards an A kind of percept, well, you'd use this electrode. If you want to bias towards a B kind of percept, you'd use that electrode. However, if the resolution you want to attain is larger than your stimulator density, and again, it's not the number of channels, it's the stimulator density that's the thing that matters here, well, then you have a bit of a problem, because if I fire this electrode or if I fire this light, I'm going to hit several different incompatible percepts. Right? So people have made some progress when in those areas where this is the resolution that we actually have. The density of actuators matches the density of what we think the neural tuning code is. Even then, though, they're usually proof of concepts that ignore, for example, that there's actually fine gradations, differences between neurons even when they're nearby. So all of this sets up the problem that in these kinds of animals going out in the world, exploring their environments, trying to get information from it, we want to get in there and sort of artificially change the neural activity patterns in some way that sort of causally tests what we think the neural models for sensation are. We need to at least take on this notion of somehow having common inputs that are nevertheless driving independent activity in different populations of neurons. And I want to stress here at this bottom that this problem is really part of the reasons why I'm so interested in it. It combines questions from neuroscience, which is, well, what is the resolution at which we need to activate these neurons? What do we actually need to do to create a percept? And that is, what would now be called causal neuroscience as opposed to correlative neuroscience. Give a stimulus and see what the neurons do. The idea is to make the neurons do something and see how the animal responds. That's how, what kind of resolution we need to do that is really still unknown. But then there's also this engineering piece. Suppose I have an idea of what kind of resolution I want. How do I actually attain it? So we've made some efforts in that direction. I'll tell you where we are. The basic idea starts with some very simple models, and then I'll come back to this model specification issue in a moment. So we take the integrate and fire model, which I imagine is largely familiar here, but it's basically a passive RC kind of charging. So you're really just looking at the charging of the neuron to threshold. In our case, we have an input, which is a conductance input. And the one thing that's going to matter here for that underactuated problem that I was just talking about is this G here, the thing we pick, doesn't have an index on it. There's one light source for all the different V's. That's the thing that makes this problem difficult and the thing that we're really focused on. And then all the nonlinearities get pushed into this reset. If I manage to charge the threshold, I get set back to rest, and so on. Right? So in this model, you can, in fact, take, say, two neurons which have this common G and nevertheless drive them to different spike patterns. I'm showing you a simulation example here where there's a red and a black voltage, and I get A, B, B, A, B spikes from these neurons using this input G down here, which is basically one very sharp, short pulse and then one sort of muted, longer pulse. So I want to describe a little bit of, how does this come about? What's the issue with underactuation? So if I had a different G for every neuron, I could look in the phase space of voltage of neuron 1 versus voltage of neuron 2, and I'd be able to push that voltage in any direction I wanted. I just pick whatever G I want for either neuron. The fact that there's a common G means, in fact, at every moment in time, there's really one sort of quasi-steady state, which is what I'm indicating with this dashed curve. So for any choice of constant G, the system wants to move to this curve. Right? That's what's going on. So how do I get different spikes? Well, spikes occur when I hit thresholds. So the B neuron will spike when I hit this line, and the A neuron will spike when I hit that line. So the thing that I need for controllability is not only to know where this curve is, but what the curvature of paths are as they're trying to get to that curve. And this example here looks like this in the phase plane. Basically what's going on is if I pick a sort of small input here, well, the curve will cross this line, but it will never get to this line. So I definitely get a spike at a neuron 2. If I push my G all the way up here, I get a curve that will actually hit both thresholds, but it hits the one neuron or the A neuron threshold first. And that's how I'm able to get independent spikes out of these two different neurons. Right? So this is just sort of basic idea of underactuated control. You can't have all possible paths, but you can still, in some cases, find paths that do hit your individual targets. And one thing that I'll also come back to in a bit, but I want to point out now is, note that our control objective is not to hit a particular point in phase space. It's just to hit this line anywhere or hit this line anywhere, because that's all that matters for extracellular recording. We don't get to see the voltages. All the stuff that Steve showed in the great modeling work that he's doing requires observation of the voltages. We don't even get to see that. All we get to see are the spike times. The only thing we know from extracellular recording is whether I hit this line or that line. That's it. So that creates some additional complications, even because of the definition of what our control objective is. But OK, so this is something we published a few years ago. And in that paper, we did some analysis looking at controllability. And so here's the upshot of that. If we imagine, so this is that alpha parameter, which is like the RC time constant, rate in this case, and beta is the strength of the input to that particular neuron. So every neuron has some different amount of what we would call leakiness or RC constant and some different amount of sensitivity to the input. If we pick a neuron, say this guy in black, we look at all the other neurons that under our particular way of doing things meet something we found as necessary and sufficient conditions for controllability, and it's this gray region. So of course, because we're giving common input to two neurons, there's no way we're going to be able to have pairwise controllability over all parameters, right? Your starting point has to be calibrated here, right? We're not starting from, hey, we're going to have some strategy we're going to control everybody. Just the fact that it's not zero, that we can do better than synchronously activate neurons is already progress, right? So, something like 25-30% of the plane seems pairwise controllable. And then you can stitch a bunch of different neurons up as long as each one of them lies within the gray regions of all the others. You can end up getting these convex subsets where here's a simulation example with 8 neurons where we create this sort of target pattern, run down them in one direction, run down the other direction. That's our target. What's shown here is at the times of the spikes of these target neurons, what's the voltage of all the other neurons, color-coded in this case. And the point is we can, in fact, get this pattern of activation through this 8-cell controllable network. Right? So it is possible, in this case, with a single input, to get independent spiking out of 8 different neurons. That's certainly better than 0. Right? So how does that scale? Well, it does, as you increase the number of neurons you're illuminating, and you assume they have some reasonable distribution of parameters, some randomness in the brain, some heterogeneity, the maximal number of cells you can control simultaneously with a single input does go up, but it goes up really slowly. Right? So there's going to be some trade-off here between how large a volume am I trying to hit where I can have independent controllability versus when do I need to add a new fiber. Right? Those are the kinds of things we want to understand better. So I want to get to some experimental results. There's more I could say, and you can look up some of the details in this paper. I'll just add on all this analysis I sort of skipped over was really just looking at the ordering of spikes without any information of timing. Since then, we've gone to do time optimal control. So can we get the spikes as fast as possible? The idea being, whatever the fastest possible way I can get pairwise controllability is, I can always add buffers to make the spike times longer, but I can never make them shorter. And so here's more of that kind of phase plane analysis, neuron 1 versus neuron 2, and there are these paths where, again, you're just trying to hit these two boundaries. We've added some bells and whistles, like some guards. If I'm worried about something, the other non-target neuron getting too close to threshold, I can just artificially pretend the threshold is lower and solve that control problem. So there's a number of things that we tried to work out here. And the point is you can still do it, even in this time optimal context, and we worked out controllability conditions. It's mostly sort of from linear systems theory, you're mostly talking about bang-bang control. You have a linear system and you're trying to drive it with this input. You just want to hit it as hard as possible in the right part of space, hit it as weakly as possible in the other part of space. Right? That's just a sort of standard systems result. But it gets a bit messy here because, again, we only care that we're on this boundary. So that creates some singularities in our cost functions and things that a lot of this paper was trying to get at. Can we solve those analytic problems? Right? So I or Shenung or Ani, who are all here, we'd be happy to talk to you more about any details for anybody that wants to dive into those things. But this is, again, in that integrate and fire model, in this model that we know is wrong. I think everyone here has probably heard all models are wrong, some are useful, right? This is a common mantra, right? So we know this model is wrong, right? Is it useful? Well, let's think about things we could do if we had truly nonlinear models. So here's an example of Hodgkin-Huxley neurons, which have been introduced for any people here that don't know them, right? And they are much more realistic, even if I don't include oxygen depletion. In this case, I'm showing you a pair of neurons where the red neuron has 10 times the sensitivity to the input of the black neuron. So it's obvious I can get red spikes without black spikes. But the question is, can I get black spikes without red spikes? Can I make the less excitable neuron somehow still spike? And in this case, I'm using a strategy that's been around in one form or another for a long, long time, the sort of pre-pulse inhibition. I give a bunch of small pulses. The red neuron will be more sensitive to those. But what's happening in this nonlinear model is that's inactivating the sodium channel responsible for the spike. So by the time I come in with my true input here, the red neuron is effectively blocked. It's already wasted all of its sodium activation, so only the black neuron spikes. So that's a strategy not available in our simple, stupid model. Right? why wouldn't we just use this? Well, in order to do this effectively, you implicitly need to track things in state space, like in this case, the potassium activation versus the membrane channel. And to get this right, you have to sort of march up this little manifold in the state space. And we certainly can't observe potassium activation. We can't even observe the voltage. All we know are the spike times, right? So this is a great strategy, but it's thoroughly inaccessible to us. So in this kind of environment, we take the attitude of, well, let's just use the wrong models. And so the kind of thing that we do is you say, well, let's take some Hodgkin-Huxley-like pair of neurons. Let's decide when we want them to spike. Using only the spike times from the plant, we'll fit the integrate and fire model. Then we'll design the control in an integrate and fire model, which will be exact, so we can get what we want, up to some details we can get into later. And then we'll apply that control back to our original true plant, right? This is a classic kind of way of thinking about this, and see what we get, except that everything's nonlinear here, so we can't provide very good, precise estimates of how well we'll do, but we can at least test it out in simulation. And here's an example of that where, as you expect, you get spikes that are okay. They're not perfectly timed, but it does kind of work, right? So this is our down in the trenches, we're trying to make things happen kind of attitude towards doing this neurocontrol. And again, the thing for us, as opposed to what Garrett just did, which was, as he said, a single neuron with single input, there's some other great work from Andre Longten's group. There's stuff from Liam Paninsky. There's a lot of neurocontrol that's in this kind of space. But to our knowledge, very little of it has really dealt with how underactuated things are. And again, in deep brain stimulation, you're not really underactuated, because what you're observing is the mass activation of an enormous number of neurons, and you're looking at the field, right? So this is what I mean about, for people here, I would love to talk more, but people are really into the details of how to formulate control problems on when what does it mean when you're controlling a one-dimensional variable, like an oscillation in a local field potential, that you know is coming from a bunch of individual neurons that are the ones that are actually stimulated? This gets to the black box kind of stuff that to which Steve was alluding, right? Is that a one-dimensional problem? Do we need to care about all those degrees of freedoms underneath, right? That's a thing that neurocontrol in general is really struggling with. In our case, though, for the sensory stimulation kind of approaches, everything is very transient. Unlike the stuff that you saw, we're not looking at firing rates that we're trying to modulate on second time scales. We're trying to do things at 10 millisecond time scales. So that's why we're very pulsatile. We're trying to get fast activation of specific neurons spiking in specific timing patterns, or at least in certain orders, right? So it's a very different kind of neurocontrol problem than trying to, for example, hit a steady state. And so maybe closed loop will help, and I'll come back to that in just a second. But a lot of things are happening too fast to use closed loop control, because the only thing we get to see are the spikes, and we're only going to get 1 or 2 spikes per input anyway. So the best we can do is to say, well, our last input sucked. Let's try to update our current input. But we can't do anything during the input that we're actually giving in that moment, because everything's over before we can even estimate the state of the neuron. Okay? So these kind of issues, maybe they come up more in questions. And again, I'd love to talk to people here about how to properly formulate these things. But let me say, this is the simulation work that we've done, again, motivated by what I think we need to do in my lab in real mice. So let me finish up by talking about real mice. So again, this is that picture of the brain, lots and lots of light-sensitive neurons. What happens when you shine a light? Well, here's an example, laser pulse, and this is about 4 different recordings overlapped. And this is what you actually see from extracellular recordings. You get this filtered out sort of background hash, and then you get some number of spikes which you have to identify in real time and figure out who's who and do all those kinds of things. So what I'm about to show you is done in anesthetized mice, not the mice running around, because we need the biophysical control just not to be completely overwhelmed by the experimental variance. Even when you have identical conditions, you of course get slightly different responses on different trials, right? But are we at least anywhere close with our kind of very simple integrate and fire mindset? So if you remember this figure, it says that something like 25-30% of neurons should be pairwise controllable if anything like this integrate and fire model is correct. So we have a way of estimating that, which is for the old school physiologists to look at strength duration curves. And basically, if the strength duration curves cross, so strength duration curve is for a given duration of stimulus, how strong does it need to be to just get a spike? That's what the strength duration curve is. I'm showing you about 75 different strength duration curves from neurons we've recorded in anesthetized mice. And if they cross, in a way I'll elaborate in a bit, that means I have pairwise controllability, and something like 27% of these recorded from real neuron strength duration curves happen to cross if I just pick them randomly. So that at least looks I lost my pointer, something like this. All right, so what's the strategy? Well, let's start with the simplest possible thing. If I look at these strength duration curves, right, again, what does that mean? It means a stimulus down here will be long and small, so kind of fat and short. The stimulus here will be short but much stronger. Right? So I look at an individual neuron, and maybe I record a curve like this. So any stimulus anywhere along this line will give me a spike. If I get some other neuron that's in red here, I can get it to spike with a slightly different curve. And so if you think about, for example, a pulse down here, it's above the one curve and below the other. So this ought to get a blue spike without a red spike. Over here, because they cross, this ought to get a red spike without a blue spike, right? So hopefully I can just find these points in space. That's what we're looking for. So here's two neurons. These are overlapped waveforms. So this is what we see extracellularly, and this gives us a sense of these two neurons recorded on different electrodes, so we have no concern about isolation in this particular case. And what ought I see if the world is perfect? Well, if I'm trying to get this blue one to spike, I'll mark that with an X. And what I would hope to see is that every time I apply the X stimulus, let's say it's the fast, strong stimulus, I'll see lots of spikes to each. The fraction of pulses that get me a spike out of this neuron will be very high. And in red here, the fraction that get a spike out of the red neuron will be low. And then if I apply this O stimulus, where I'm targeting the red, I ought to see the opposite. Now, I don't expect to actually see this, because I know that I have this underactuated noisy problem. It's not going to be that good. What do I actually see? Well, in an example here, you can see that, well, here are the X's, and the blue's high and the red's low, and here are the O's, and the red's high and the blue's low. So it's not perfect. We're looking at 60% instead of 100%. But we do actually get this pairwise control. So let's be clear. Two neurons in a bath of all kinds of other neurons receiving synaptic inputs and messy and everything that you don't want to have to contend with on a model is all there. But we are able reliably to get more spikes out of one neuron than the other, or flip that, even though they're both being stimulated by the exact same light. And we're doing it all through temporal modulation that exploits the heterogeneities. Now, what are the problems? Of course, over time, these things drift, the kind of state switching that Garrett was just talking about. Drifts in the electrode, and also the fact that we're stimulating changes the state of the brain too. Right? So we have all of these things to contend with. We're now working through a kind of adaptive approach that maybe can deal with this stuff a little bit better. The point is, there is some degree of controllability. It's not great. These are the kind of problems that we've been working on very hard, and these are the kind of things I would love to talk to people here about in more detail. But so just to summarize all that up, we have this issue from sensory prosthetics. If we're thinking about those kinds of problems, or really just any problem involving injecting information in the brain where our theories tell us that the identities of different neurons matter, and we have to get these multi-neuron spike patterns if we hope to actually create function at a neural level, but our stimulation technology is just not up to the challenge as it exists now. We are making better hardware. Things are getting better. There are some proofs of concept where this works, but we're trying to also improve the software. We use the kind of buzzwordy way of describing things so that we can get more out of each channel that we have in there. And this is both an engineering problem and an interesting neuroscience problem, because of course the whole reason we're using this is to actually then make the nervous system do something and say, did the animal think that it touched something that wasn't there? Can we remove the percept of touching something that was there? And in the limit, ideally, actually start to make real technologies that replace function in patients, although certainly from our work, that's off on the horizon. So a lot of people contribute to this work, especially Sam Brown and Zhang Jinyang in my lab on the neural recordings. And this is all in a long-running collaboration with Shenung and colleagues here at Washington University, and a lot of funding as well that we have to appreciate. And I thank you for your time.
Time for one or two. John, can you get the mic?
Okay, so
Please go ahead. Please go ahead.
What?
Yeah, please proceed.
Okay,
Yeah.
Jason, I have a question. So, you're doing these things that have to do with sensory motor control in the rat, and he's moving his whiskers, and you're trying to be a little bit disruptive with this stimulation. Now, the neurons that are firing in certain patterns, and these are spatio-temporal patterns, I assume, in the brain. Right? I'm just asking a very naive question, because I don't know much about this. But the spatial temporal patterns may be fairly sensitive to the order in which neighboring neurons fire that are responding to the notion of the whiskers. And if you're doing this optogenetic stimulation, are you perhaps, just disrupting that pattern in a way that's very unnatural, that wouldn't ever occur in just natural sensory motor?
Mm-hmm. Yeah, so the basic question is sort of, are we actually stimulating in a way that matters to the neural code, or are we just getting in and pounding on the guy with hammers? And for the experiments I showed you at the beginning, hammers all day. Absolutely clear, right? We're coming in at a certain time and saying we're adding spikes or we're deleting spikes. Now, where we may be able to get away with it is because we're talking about problems that are dominated by contact. The animal's whisking around in space and hit something, and maybe that's sufficiently sort of ballistic that our little pulses are at least within a space where it's going to respond to it as something other than just that was weird, right? Also, there's some details with the optogenetics you can kind of dial into particular cell classes and for the experts, in particular layers, which I think matters quite a bit. So there are ways to exploit who's labeled virally that maybe is a little better than what I just said, but in general, I see it as sort of these two railroads that I'm trying to get to connect somewhere in the middle of the country. There's what we're doing in the freely moving animals, where the behavior alone is so complex that the chance to get in and do really careful biophysics is just not there. And then the anesthetized animals, where we're trying to improve what we can do biophysically, but we don't have those two things connected yet. That's absolutely the case. What's interesting is that the animals will still sometimes respond to this horribly crude thing we do in ways that don't seem totally random, and I didn't show you at all, but all of that first stuff is actually set up for the experiment we're doing now, which is the thing I'm super excited about. I'll just say this quick and then you can yoke me off the stage, which is those water rewards I showed you are under robotic control. So in particular, what we have now is a system where we use non-uniform statistics of where the water is. And so the animal learns in its environment water is more likely to be over here than over there, and it starts to develop these expectations. And then that means when we go in with our probe stimuli, we can try things like probe when water is likely versus when it's not likely. And we're trying to sketch out this function of, well, how well do we fool the animal, right, based on this sort of map of what it ought to expect. And the reason this matters as an experimental design is because a lot of people will show that they can successfully drive an animal to respond to a stimulation, but that actually isn't very good behavior in my opinion. This is the thing we'll talk about offline, I guess. But what you actually want to do is fool animals because they have no motivation to search in the wrong place where there isn't water. So that's in some sense a better test that you're actually fooling them. And that's what we're doing with the experiment that I just started to sketch out. So we're working towards it, but I freely admit we are not there now.